All right. Good morning, everyone. And this is going to be the last of the remote lectures I'll be posting for lab. And really, this particular lecture itself is not necessary for class because even if we have regular labs, this actual handout is mostly supplemental information at best, and only a select amount of information from here would have been given to you on the quiz. Now, regarding the quiz, I'll be uploading a quiz today, and I'll also be posting about this on Canvas, this information on the quiz. And the quiz, I'm going to give you basically a whole week to do it. It'll be a very homework, like basically a homework format quiz. So open notes, there's no time limit. However, the only caveat, there is going to be only one attempt on the quiz. So make sure that you spend a lot of time on doing it so that you answer every question correctly. It's going to be mostly multiple choice. I think it's going to be only multiple choice. So just it's quicker for me to grade them. Well, anyways, without further ado, so the actual last 14 week, the last 4 weeks well, really the last 5 weeks of lab, and everyone's been doing it, is just a lab report. So, and if we would have actual lab without, if there was no COVID-19 infection, a pandemic, then basically every lab from after your midterm, your practical exam, would have been basically the first hour would have been some exercises, and then the remaining 2 hours or so, give or take, approximately, would have been you guys working on your unknowns. So you'll be doing all different experiments. Now, the reason why we this lab as well as the upper level lab where we give this particular supplemental handout lecture is because sometimes you would need to do some of these tests for some unknowns. Some of the bacteria could be a little bit more difficult. However, for this particular situation, the bacteria, the unknowns I gave everyone, none of them are going to be really utilizing any of these tests, fortunately for you guys. Okay? Not most for the most part. Okay. So a lot of these tests you don't have to worry about. Okay, but I'm going to go over some of the key ones. The rest of them is mostly going to be for your own knowledge. Okay, if you want to read through them, you go right ahead. Okay, they're pretty insightful, some of it. But I'm just going to go label some of the really key ones. Okay, so without further ado, the first one I'm going to look at is the nitrate reduction test. Now, I have a whole figure here that I'm not going to go really into too much because it's not really important for you guys. This is more complicated and it's more uh, a lot more biochemistry that you don't really need. And you guys could email me if you want me to talk about it more. And I could, give you a nice response on it if you would like. But basically, nitrate groups can undergo reduction and oxidation, okay? And they exist in different states, okay? Now, if an organism is able to reduce nitrate, so it's going to be NO3 minus, so that's nitrate, into nitrite, NO2 minus. So when you reduce something, that means you're giving away an oxygen. You're losing one oxygen. That's what reduction means, okay? You're losing one oxygen here, okay? And it can reduce nitrate into nitrite and then nitrogen gas, okay? Such as well as ammonia, okay? Nitric oxide, nitrous oxide. So it's just the reduction of nitrite, okay? Different nitrites, all right? Oh, the nitrite, nitrate into nitrite, nitrogen gas, or maybe other compound, okay? You're just looking for the reduction, okay? Now, how does this happen? Well, you have a lot of most nitrogen-reducing bacteria will have something called nitrate reductase. Nitrate reductase, and I'm going to actually underline it right now with my marker here. Nitrate reductase is an enzyme that will catalyze the reaction to reduce nitrate into nitrite. Okay? Now, the test itself, when in lab, we use a broth. It's called nitrate broth. And nitrate broth has something called potassium nitrate. Okay? Has potassium nitrate, has beef extract and peptone, as well as an inverted Durham tube. Okay, so you have a Durham tube inside of it, and everyone remembers what those are for, right? This is for detecting gas production, right? From the fermentation assays. Now, if gas is found, so this is what happens with where we use the Durham tube, okay? If you find gas production to be found in the tube after incubation, and the organism is known to be a non-fermenter, okay, and this organism it doesn't is a non-fermenter, that means there's no sugar being fermented. This will then mean denitrification took place, okay? When you denitrify something, you're going to be producing nitrogen gas, okay? It's actually shown in the figure below over here, and I could quickly go over it. Okay, right? Now, if you don't see any denitrification occurring, meaning that there's no gas production in the Durham tube, okay, then after that, two reagents are going to be added, okay? And the two reagents that are added is sulfanolic acid and alpha naphthylamine, okay? And this is going to be testing for the presence of nitrite, okay? Now, when you add these two reagents together, so the sulfanolic acid and alpha naphthylamine together into this test tube after the incubation period, if a pink-red color forms after the addition of the two, then that means that there is nitrite present, okay? And if nitrite present, that means reduction occurred, okay? Because to get nitrite, you got to reduce from nitrate first, right? It goes from nitrate to nitrite. That's what reduction does, okay? Now, if no reaction occurs, meaning a pink-red color does not form when you add these two reagents together to the tube, then you add one more reagent, okay? You're gonna add some zinc powder, okay? And this is gonna be like the double check to make sure there really isn't any nitrate, okay? Now you're checking to see if nitrate is present, okay? And if there's nitrate present in the broth, this means that no denitrification occurred, okay? So zinc powder will turn red. So if nitrate is present, the zinc powder will turn red, okay? But if there's no color change, it remains a colorless solution, then uh, then that means denitrification to ammonia was happening, okay? So if you have denitrification to ammonia or some other organic compound, that means that the zinc powder will not turn red. It will just remain colorless, okay? And I'm not gonna go through the procedures because it doesn't make any. I mean, especially for the lab reports, none of you guys will. I don't think anyone had to do this, or maybe there is one. I don't remember. Uh, but if you need to actually use this on the lab report, like the procedures, I would definitely refer to this for the material methods. Okay, if it's applicable. Okay. Now let's just take a look quickly at the ammonia, the nitrogen cycle. Okay, and this is going to give context. Okay, zoom in. Can I zoom in? I could zoom in. Very nice. Okay, so all right, so we're going to start from NO3. So nitrite. Sorry, nitrate. Okay. Now nitrate, when it's going to undergo nitrification, meaning it's going to be reduced, it reduces into nitrite. Okay. All right. So, and nitrite, okay? Now, nitrite then undergo denatrification, okay? Sorry, either go denatrification or nitrification, okay? If this becomes, if it nitrificates, it's going to become ammonia, okay? If it denitrificates, denitrification, it's going to become nitrogen gas, okay? And this is where the Durham tube comes into play, okay? The Durham tube is going to detect the production of nitrogen gas, okay? Now, don't worry about all different things. These you don't have to worry about this. Okay, that's a little bit too complicated. But there's different pathways nitrate and nitrite could take, and they could form NH2 as an intermediate. Okay, and then as an intermediate, it could then become ammonia. Okay, all right. So this is kind of like the general scheme. I'm not gonna test you on this or quiz you on this particular graph, this chart. I just brought it up there. In lecture itself, I kind of generalize it too. This is more for the upper level material, and because these notes that I made are both for your guys' level and also the upper level. But there's no real point for me to make two separate notes, but because the procedures are the same thing. So a lot of this is going to be supplemental information if you want to go through it. Okay. However, I will want you guys to know these definitions here, and I'm gonna put them into brackets right now. Denitrific or microorganisms. they could transform nitrite into other forms of nitrogen, such as nitrite, the gas, nitrogen gas, nitric oxide, ammonia. Okay. You don't have to worry about this chart because I won't actually even ask you guys to label this. Okay. And to be honest, it's not gonna be a problem because it's all open notes. Okay. So you can just directly refer to these notes anyways. Okay. You have to know what nitrifying microorganisms are. Okay. That means they can oxidize ammonia into nitrite. So oxidation is gonna be the reverse of reduction. Okay. So if you're gonna be reducing NO3 to NO2, oxidizing NO2 means that you're gonna go back to NO3. Okay. It's just the reverse reaction. Okay. So let me just get rid of this, a little bit too cluttered. So this means that let's say you want to oxidize. Okay. You want to oxidize NO2 to nitrite. If you oxidize nitrite, it becomes nitrate. Okay. If you reduce nitrate, it becomes nitrite. Okay. If you reduce nitrite, reducing nitrite then is going to become ammonia. Okay. If you want to oxidize ammonia, that means it's going to become nitrite. Okay, it goes back. It's reversing. Okay. But yeah, that's basically for this. Right. Now I'm going to show you the test itself now. And these are the three outcomes, okay? And we'll go through them. Okay, so these are the different possibilities that can happen if you do the nitrate reduction test, okay? Now, the one thing I will tell you right now, in these tests, you don't see the Durham tube. That's okay, okay? But I will want to see the major color changes, okay? So here is a negative control, okay? That's negative control because there's no bacteria there, so it's always going to have it's going to test for a negative reaction because there's nothing growing there, okay? So this is how a negative control looks like. So this is going to be like a standard, okay, that you would use normally, okay? So let's see what happens, all right? So here, if you don't add any zinc, if it turns color, if the color of the broth after you add the two reagents into it, if it turns color to a reddish brown, like this, okay? If you remember, let's go back. Let me go back quickly. It's a shame that you got to turn off the, go back up here, okay? Let's see here. Where are you? If you okay, so you add stages. Okay, so phenolic acid and alpha naphthylamine, okay? If you see a pink red color, that means it's a positive reaction, okay? If not, you're gonna have to add zinc. Because if you don't see anything happening, you're gonna have to zinc. Okay. If you see a red color form after the addition of zinc, it's a positive reaction. This is a little bit it's an annoying test. Sometimes it doesn't always work. Okay. But over here, you can see that the actual bacteria here, it turned kind of reddish. Okay. All right. Now, let me see here. Sorry guys, I'm just fixing this right now. This thing is come on. There we go. All right. Sorry about that. So over here is going to be positive with zinc, and over here is going to be our negative control, okay? Or negative, I should negative control, but it's going to be our negative outcome, okay? This means that for nitrate reduction, it's negative because when you add the zinc, when you add the two reagents and the zinc together, there is no, actually, let me see if I get better pictures than this. I don't like these pictures. I'm sorry, guys. Let me just take a look quickly. Yes, I'm using Google, guys. I don't have any in-house example. I apologize for this. Okay. So if you add zinc, okay, let's say there's no color change and you want to add zinc to it, you want to check to make sure there is reduction happening, right? If there is a change if there's a change here of color into red, that means that it's a negative reaction because there's we still have a negative we still have the nitrate. The reduction is happening. Is not happening. Okay. Now, if you don't have any color change and it remains like to be like a yellowish, like a bacterial looking color like this, okay, that means that nitrate is reducing into nitrite or maybe ammonia. Okay. I'm not gonna go too much into detail with the like I said with the biochemistry, but just know that this means after the addition of zinc, this means reduction is happening. This means no reduction is happening. Okay? That's all you need to know for that. Okay, anyways, with that being said, let's go to the next thing that I want you guys to know. Okay? So all these tests here, all these different differential tests with hydrolytic enzymes, I'm only going to tell you which ones you need to know. Okay? A lot of these, most of it's supplemental, and you guys can read through it. I have pretty detailed notes here that I have for you guys. Okay? That I made. Now, the first one is called the starch hydrolysis test, and it's also known as a starch agar plate. Now, this one here, you're looking for the bacteria's ability to hydrolyze starch. So that means that it's going to be secreting certain enzymes that are going to break starch apart. Now, normally, bacteria is not able to break down starch because it's too big to pass through the cell wall. So what it does is it'll start to secrete certain hydrolytic enzymes that will break down. So the actual enzyme is alpha amylase. Okay, so it's going to be breaking down starch into glucose. Okay, so alpha amylase will break down. You don't have to worry about the 1,4 alpha glycosidic bonds, but just know that alpha amylase will break down starch into glucose, and glucose itself will be able to be taken in by the bacterial cell wall. It's able to travel. Okay, so how do we actually test for this? Well, the actual starch agar plates, they appear black. Now, why do they appear black? So if I have to show you a starch plate, and I'll show you actually in a second. If I show you an uninoculated starch plate, it looks black. The agar itself is black, and the reason for that is because one of the components in the starch agar plate is iodine. And iodine, what it does is in the presence of starch, it interacts with it, and the interact between iodine and starch produces this very black color. Okay, all right. So, and I'll show you what happens. Okay, let's see here. Right now, what I'm talking about. Come on, where are you? There you are. Okay, so I'm going to show you right now. So this person here, and again, this is from Google. I don't know. This is not me. All right, but the person inoculated two different bacteria. Okay, bacteria number one is over here, and bacteria number two is over here. Okay, come on, there we go. All right, now this area here is not inoculated. This area here is not inoculated. Okay, so this is how the plate looks without any bacteria. It's just black. And this is because of the interaction between iodine and starch. Okay. Now, how do you know if a bacteria is synthesizing and also secreting alpha amylase? Well, let's look at this one. Okay. If the bacteria is going to secrete this amylase, alpha amylase, it'll start breaking down the starch into glucose. And when starch is broken down, the black color goes away because it's taking away the starch that iodine would interact with. So you're taking away that interaction. So once you take away that interaction, that black color goes away. All right. So if you look at the one I just drew an arrow towards, you see there's no formation of any halo effect. There's no, like, the color of the black agar remains the same. Okay. There's no color change of the agar. Okay. So this here indicates a negative reaction. Okay. So this is negative alpha amylase. Okay. Now let's look at this one here. Okay. On the right side. Now, if you notice, okay, so this here in the middle, this is the actual bacteria growing. Now, if you look at over here, okay, this section over here, make sure I'm drawing this. That's a bad line. Sorry, guys. If you are to look over here, okay? Oh, good Lord. If you were to look around here, okay? It's a little halo, and it goes around the entire around the perimeter of the bacteria, okay? This happens because the bacteria is secreting, and it's synthesizing and secreting alpha amylase, and it's going to be breaking down all the starch around it, okay? Once you break down the starch, you're going to have the appearance, you're going to get rid of that black color because you're taking away starch, okay? It's hydrolyzing starch, all right? So this halo effect means there's no starch, and this indicates a positive reaction, okay, to the test. So this means that that whatever this bacteria is over here, this one here, is making alpha amylase. Okay, that's all you need for that. Okay, you have to know that for the quiz. Okay. Oops. Okay. Now let's look at the gelatinase test. Okay. Now the gelatinase test, guys, is another hydrolytic enzyme test. But this time we're looking for the gelatin, the presence of gelatinase. Is the bacteria able to synthesize gelatinase? Now, what gelatinase does is that it actually is going to break down gelatin. Okay, and gelatin is a protein. So let me just underline this for everyone. So it comes from collagen. Okay, so right here, gelatin is a protein that comes from collagen. Okay, usually you find them in the vertebrae connective tissue. So you can imagine that if you have a bacterial infection of a bacteria that's able to produce gelatinase, that means it's going to be able to break down the connective tissues around your vertebrae, right? That's what it means, and that's bad. Okay. Now, well, what happens? So a bacteria that's able to produce gelatinase will liquefy a medium that's saturated with gelatin. And I'll show you. So you basically, the test is going to involve you, uh, quote unquote, melting gelatin. You're liquefying gelatin. Okay, that's what you're looking for, the liquefaction of gelatin. Okay, so this means that in a tube that's filled with gelatin, jello basically, if a bacteria is able to produce gelatinase, it's going to liquefy it. That's a positive reaction. Now, if the gelatin remains solid, that means that the bacteria is incapable of producing gelatinase. Okay, and this particular test is very good for distinguishing Staph aureus and Staph epidermidis. If I recall, I do recall for some of you guys, a little hint for some of you guys. Uh, I did include this test, okay, and it's very good for distinguishing Staph aureus and Staph epidermidis. And in lab, like and these are usually classical unknowns that I give traditionally to the class, Staph aureus and Staph epidermidis. And this is going to be a test you need to do to be able to actually narrow down whether or not it's aureus or epidermidis. Okay, and it's also useful for distinguishing Serratia and Proteus species from other Enterobacteriaceae family members. Okay. And I'm going to show you right now how this test looks, okay? So we're here, there we go. So, so the way this works, the way you actually do this test is you're going to inoculate the bacteria into these tubes, these test tubes, okay? And so they appear, first of all, they come like this. They come solid, which makes sense because they're gelatin, all right? And then you have to use the needle that has bacteria on it, and you're gonna puncture the middle of it with the needle, and you inoculate the bacteria that way. Throw it in the incubator for 24 hours overnight, okay? Then just a side note, guys, okay? When you actually remove it from the incubator, it would always come out melted like this. So don't, like when you actually take the tubes out of the incubator, don't just say it's positive for gelatinase because the 37-degree temperature and higher is always going to melt the gelatin, okay? It actually denatures gelatin, okay? So what you need to do to confirm that you actually broke down gelatin is that you got to throw it in the refrigerator for like 5-10 minutes, maybe longer. It depends. Like I've winged it to reduce the amount of time, but you could leave it for a longer period of time. You could leave it for 30 minutes if you want. right? Nothing bad will happen. Okay. But basically, you leave it in the refrigerator, and what you're doing is you're basically trying to see if it's going to re-solidify again. Okay. It's kind of like Jello. It's literally like Jello, right? If you place Jello into a hot incubator, the Jello is going to liquefy. But if you place it back into the refrigerator, it should re-solidify again. It's the same exact principle as making Jello. Okay. So you place it back into the incubator, you take it back out. I'm sorry, but you back into the refrigerator, you take it back out of the refrigerator. Now, if the liquid remains, if the media remains liquefied like this on the top portion, this indicates a positive test for gelatinase. This means the bacteria has gelatinase. Okay, it's producing it. Now, if it becomes solid again after refrigeration, if it reverts back to being a solid, that means that there is still gelatin present in this media here, and therefore it's negative for gelatinase. That means the bacteria is negative for this test. That's all you need to know, and you got to know this, okay, for the quiz. All right. What else do I have to explain here? Oh yes, last but not least, this is the very last one I want to go over, and this is gonna be the DNase test. Okay. Now, the DNA test is looking for the ability for bacteria to produce DNase, okay, which is deoxyribonuclease. Deoxyribonuclease is very important for breaking down DNA, okay? So it hydrolyzes DNA, okay? And it's going to be the same principle as the starch test, where you're basically looking for a halo color to form around the growth of the bacteria. The only difference is the color of the actual agar is going to be a dark green, blue colored agar plate instead of a black plate. Okay? And I'll show you right now. Okay? So, and of course, it's going to, so DNA remaining in plate will be visualized by acid precipitation, turning the plate in opaque white color. Okay? So the actual, and I'll show you right now. So it's going to be because of the acid precipitation that this change occurs. Okay? And I'll show you right now. So, okay, so this is a negative test, okay? And you can actually see the color of the agar, okay? So the actual agar for the DNase plate is dark blue, a dark green color, okay? It appears dark green, okay? Now this person made a little bacteria patch over here with Staphylococcus epidermidis. Now Staphylococcus epidermidis is known to test negative for the production of DNase, okay? So if you could see here that the agar itself has no color change. There's nothing changing about the agar, okay? That means this is negative. This is a negative test for DNase, okay? It's not breaking down any DNA and causing acid precipitation, okay? So this is a negative test, all right? No color change on the agar itself, all right? We're not looking for the growth. Now, if you don't see any growth of bacteria on that just means that you didn't give any back. Either you tried to inoculate dead bacteria, so maybe when you flamed your metal loop, it was too hot and you killed the bacteria upon transferring into the plate, or you just didn't take any bacteria at all from your slant or something, okay? All right. So if you ever see no growth on this, it's not a negative test. It just means you did it wrong, typically. Okay. Now let's look at a positive test for this. All right. Here we go. So do you guys see how light the color became, and especially immediately the perimeter around the growth of the bacteria? You see how you got that halo effect around it, a clearing of the greenish blue color. This means you're going to have some acid precipitation. And this acid precipitation is going to work by creating this halo effect. So this halo effect will it shows you that the bacteria here is actually producing and secreting deoxyribonuclease, aka DNase. And it's hydrolyzing DNA. Okay? So this will be a positive test for DNase. Okay? All right. And that's about it, guys. The rest of this stuff here, you could read it for your own knowledge, but I don't normally go over this in detail to begin with. This is mostly supplemental information for some unknowns you guys may encounter. And I actually bring over my big lab manual into lab, every lab, to actually have a reference guide for you guys to look through. Luckily, I didn't choose any bacteria that you really need to use these for the unknowns, so don't worry about this. But focus on the ones I've mentioned that will be quizzed on. Okay, the quiz will be very easy. All right, and that's about it, guys. Oh yes, the one thing I should mention. Okay, with the procedure for DNase, you actually need to do one thing after the addition. Usually, what you do is after you grow it for 2 days, you have to flood the plate with hydrochloric acid. Now, hydrochloric acid that we use is 1 molar, so 1 molar is pretty corrosive. So you want to get that, stay that, keep away from skin contact. And also, when you're handling 1 molar hydrochloric acid, don't smell it. It smells horrible, and it's unpleasant. It's a disgusting smell. It can actually cause some harm, okay, to yourself if you inhale too much of it, okay? And so this is important for the quiz to know. It's a corrosive. You have to add some corrosives to it, okay? And if it's positive for DNase, it's gonna have a halo. If there's no DNase, then it's negative, okay? If there's nothing, no halo being produced, okay? That's a little detail that you need to know. Okay, that's it, guys. And this is the last uh, recording